So the first one is a talk on targeted therapy by Dr. Scott Woodman, who you met this morning. And as I mentioned this morning, Dr. Woodman is a medical oncologist at MD Anderson, and he's specializing in some of the rare melanomas, and particularly uveal melanoma. And will talk to us today on targeted therapies. And so, Rob, will give you the 5 minutes.
Yeah.
Okay.
Great, thank you. Okay, thanks again, Sarah. And I have to say, as I said at the beginning of my last talk, it's really special and meaningful to be at this with the patients, and the patient and caregiver talk was just really phenomenal. And I feel honored actually to be a part of this. I'm at MD Anderson. I work in the melanoma medical oncology group as well as the systems biology group. And my disclosures are that I receive research funding from GlaxoSmithKline, Bristol Myers Squibb, and I am a consultant for Life Technologies. All the drugs I'll be talking about today are off-label in that there's no approved therapy for uveal melanoma, as you know. So what is targeted therapy? Actually, I think I'm going to use this arrow. It might be better. Yeah. So what is targeted therapy? Historically, patients were grouped pathologically. That is, what does the tumor look like under the microscope, and where did the tumor arise? And so then in phase 1 trials, if patients were given a drug and there were no responders in the phase 1 trial, that drug would often be stopped from development. In other phase trials, if you had responders and non-responders, you didn't know why. And just as was being said earlier by Dr. Sato, is trying to figure that out is very important. We want to spare the people who are not going to respond and really point and target the therapies to folks who will respond. So we think we're in the molecular era now. As I talked to you earlier today, we now know a lot more about these tumors. And so picture these greens and reds and purples being different molecular profiles. And this really helps in the phase 1 setting because we can enrich trials with patients that have molecular markers that we think will respond and thus allow for drugs to be further developed. In fact, the drug Herceptin for breast cancer would have never been pushed forward and approved because it requires the patient to have the HER2 a new receptor, and so likewise in other cancers. And then in the treatment of disease, that the responders will probably have similar molecular markers that correspond with the particular drug that was given. And this is proof positive in that the most effective targeted therapies for advanced cancer are linked to biomarkers. The poster child for this is CML, in which Imatinib, which you may know by the name Gleevec, was used. And in GIST, a gastrointestinal stromal tumor, Imatinib as well as other drugs are also used. This happens to have a c-KIT mutation. For these patients, in the chemotherapy era, they had a 7-month median overall survival. In the now 1-pill Gleevec era with GIST, it's nearly a 7-year median overall survival. So it really shows you the power of if you have the right target and the right drug, potentially you can really make great gains. And then, of course, in this past year, 2 drugs have been approved for cutaneous melanoma, Vemurafenib, which targets BRAF mutations, and Ipilimumab, which targets CTLA-4. And I'm going to talk about both of those in just a minute. So melanoma is not melanoma is not melanoma, and this group knows that better than any group. Non-chronic sun-damaged melanoma, so this is typically on the chest or the back or the upper arms, have BRAF mutations in about 50% of patients and NRAS mutations in about 20% of patients. Chronic sun damage, these typically happen on the face and the neck, and just as the name indicates, this is from chronic sun exposure and has low rate of BRAF mutations, NRAS, and a small number of KIT mutations. Mucosal and acral lentiginous melanoma, mucosal happening in the sinus tract, the mouth, the vaginal, anal rectal areas, have KIT mutations. Acral lentiginous on the palms of the hands, sole of the feet, also with KIT mutations. But then look at uveal. Uveal, BRAF mutations, 0. I put a question mark there only because some folks have done very, very sensitive assays within primary tissues and have found some BRAF mutations. But these have been required sensitive, as I say, sensitive methods. And also the question of whether or not, even if those clones exist, those BRAF mutation clones exist in the primary, do they end up in the metastatic? And I can say of the metastatic that we've looked at, we've not seen BRAF mutations yet. No NRAS mutations have been reported. No KIT mutations have been reported. But as I said to you earlier today, GNAQ mutations, GNA11 mutations make up 85% of primary tumors will have these, and BAP1 mutations exist in 85% of monosomy 3 tumors. So it's a very different genetic profile. So here's the examples of the point I was making earlier. If you have the right target and the right drug, we're seeing this now in skin melanomas. This is a patient who had a BRAF mutation and got a BRAF inhibitor, and this is a 2-week PET scan, if you can look at that. Really remarkable. Here's another patient of ours who had a KIT mutation with mucosal melanoma, and this was in her pelvis. This is all tumor that's active, and this was a month later, actually. And she ended up getting a pretty good response, a stable, she had a near partial response, near partial response, and then had stable disease after that. So I showed you this slide earlier just because I want to reinforce the issue that unlike these two genes that are mutated, which are activating mutations, mutations in GNAQ and GNA11 are quite different because in the inactive state, it's in a diphosphorylated form. In the active state, they're in a triphosphorylated form, and then they have this intrinsic enzyme that's represented by the scissors here that cleaves off that phosphate group and returns the protein to the inactive state. In the mutant GNAQ, that intrinsic GTPase is non-functional because of the mutation, thus always in the triphosphorylated state, thus always on. And so I showed this figure earlier. So GNAQ, if it's always on, is signaling through multiple pathways. And I highlighted a few of their what we call effectors, molecules that carry out the function of GNAQ and GNA11, MEK, PKC, PI3 kinase, and AKT. And I focused on these because these are molecules for which drugs are available in clinical trials as we speak. So rather than, earlier I talked about advances that are being made, and I highlighted the fact that the binding of GNAQ and GNA11 to PLC beta or ROGF might be intervened or interdicted upon, and that's something that is being worked on. We do have drugs at present for these mediators. Other potential important molecules in uveal melanoma is MET, or also called Cmet, and this is the receptor for hepatocyte growth factor. And because hepatocyte growth factor, of course, is made in the liver, there's some thought that, and it signals through MET, there's some thought that by blocking that interaction, and a signaling downstream of MET, you may have an effect on the tumor. Another one is IGF-1R, which is insulin growth factor 1 receptor. And again, insulin growth factor 1 is made in the liver and signals through this receptor. And both of these receptors have been shown to be expressed in uveal melanoma cells, and so there's some thought that they're very important for growth and survival. So here is a quick list, and I know it's a busy slide, but I just want to show you that, for example, for this target, the IGF-1 receptor, multiple agents in phase 1 trials, currently ongoing, and of course all of them have these NCT numbers that you're probably all familiar with. Cmet, the other receptor that's at the membrane, again, multiple phase 1 trials that are ongoing and accruing. So the drugs are in the pipeline, and thus, if one thought that either of these receptors were important for uveal melanoma, they could be placed on a phase 1 trial. Now, the MAP kinase, which is the MEK I mentioned to you earlier, there are phase 2 and 3 and 1 trials ongoing with MEK inhibitors by different companies. mTOR is downstream of the PI3 kinase pathway, as well as these other targets that are part of the PI3 kinase pathway. and multiple drugs are in development for those in phase 1 and 2. But clinical trials, particularly for advanced uveal melanoma, come in different flavors. So the one is, well, the first thing to be said is, and you already know this, that there's been 20 years or more of chemotherapy being used in uveal melanoma without efficacy. So one thought is, well, could we do it in a different way? Do hepatic arterial infusion? Could we put the drug in a liposome, which allows it to be taken up by the cell potentially better? Could we take the drug, again, put it in and stabilize it in a nanoparticle to make its delivery better? So these are all ways of modifying the way that the drug is given. Another is to couple chemotherapies with targeted agents. So here, for example, is a sunitinib plus temozolomide. Temozolomide is sort of an oral form of dacarbazine. Bezacizumab, which is a VEGF inhibitor. Genesense, which is an anti-BCL2 molecule. BCL2 is important for cell death, and so by blocking it, you can induce cell death. And so there's 2 different trials, one with triosulfan, another with carboplatin paclitaxel. So then the third strategy is really the targeted therapies. So AZD6244 is a MEK inhibitor, and this is a verse temozolomide, again, a form of chemotherapy. IMC-A12, anti-IGF-1 antibody. We're doing this trial along with Dr. Sato. And then there's, these get messy with the names, but SOM-230 plus RAD-001, which is somatostatin, which can serve as an anti-IGF-1. And then also the other is an mTOR inhibitor. So you get the benefit of blocking the IGF-1 as well as the downstream PI3 kinase pathway. STA9090 is an HSP90 inhibitor. This is a molecule that stabilizes proteins, and so potentially this could serve as an anti-Cmet. It's one of the molecules that HSP90 stabilizes, so you could in fact block Cmet as well as potentially other molecules. And I listed here essentially who's doing these trials. This is Memorial Sloan Kettering for the shorthand, Dana-Farber Cancer Center for another. Vorinostat, or SAHA, is an HDAC inhibitor, which is a histone deacetylase inhibitor. I'm going to tell you why that's a potential tool in a moment. That's being done at Memorial Sloan Kettering. And sotrastarin, which is a PKC inhibitor, and that's being done at multiple groups, but Dana-Farber and Memorial Sloan Kettering. So let me just go back, because I just went through this list. MEK, IGF-1R, IGF-1, Cmet, and PKC. So there you go. We have a drug for this in uveal, a uveal-specific trial with a PKC, and then also targeting AKT, actually mTOR, which is not shown here, which is down below it, and targeting IGFR and Cmet. So a lot of the major targets that we've thought biologically important for uveal melanoma, there are drugs available in either phase 1 or even uveal-specific trials, which is very encouraging. And then I'd be remiss, I know this is going to be a separate topic of immunotherapy, but I'd be remiss if I didn't mention the fact that there are trials for uveal melanoma that involve immunotherapies. An exciting one is anti-CTLA-4 or ipilimumab. This has been shown to have efficacy in cutaneous melanoma, and we think that there's no reason immunologically that it shouldn't have some efficacy in uveal melanoma, but that's why we're doing the study, of course. What is CTLA-4? It's a brake on the immune system. So your immune system is constantly balancing the gas pedal and the brake. And it has multiple gas pedals and multiple brakes. And one of those brakes is CTLA-4. And one way to make the immune system rev up is to block it. And so you're taking your foot off the brake a little bit. And that's represented here, an antibody that binds to it. Another molecule that's very exciting I hope gets FDA approved in the near future is anti-PD-1. Again, another brake on the immune system, and so there's an antibody that can block that as well. I should say one other thing about our trial, again, not an advertisement, but just to say that we're going to be doing it in the metastatic as well as the adjuvant setting, the ipilimumab. So it'll be an adjuvant trial as well. So MEK. Some early results with blocking MEK has shown some efficacy. This won't be reported for a little bit until the trial is completed, but there have been responders to MEK inhibition, and that's encouraging. Let's skip that. But is it sufficient? Probably not. It'll probably be the fact that you get short-term responses, and in the majority, or in a few patients, but the vast majority of patients will probably have stable disease or will have progression. So potentially another molecule to block would be the PI3 kinase pathway, members of the PI3 kinase pathway. And in my laboratory, we've actually done this. And so these are what we call spheroids. These are uveal melanoma cells that we've grown to be all together in a ball, like a tumor. And then we've treated them with MEK inhibitor increasing dose, an increasing dose of PI3 kinase inhibitor. And the white that you see, it represents the increase in cell death. And you can see that as you increase the dose of both of these, you get a synergistic effect. Because if you just use PI3 kinase alone or the MEK inhibitor alone, you see a little bit of death, but it's really the synergy of both that is marked. And that makes sense because we really believe that combinatorial therapies are going to be, at the end of the day, what we're going to need. And that's because signaling in a cell, this is a cell, signaling is complex. So here's your MEK right there, and there's your PI3 kinase, and I call this the whack-a-mole problem. Does everybody know what whack-a-mole is, right? So you hit the MEK, and then something else pops up, right, a mole, and then you hit that mole, and you're doing this, right, trying to whack the moles. Well, we believe that it may be the case that there may be essential kind of Achilles heel nodes within the signaling network that if we hit, can kill tumors and kill them well enough that we'll get complete responses. So will this combination do it? We won't know until we do it. But there are many different combinations based upon the individual drugs that I just showed you. You can imagine HDAC plus, you can imagine MEK plus, you can imagine PKC inhibitor plus, right? And I think that that's what the future will hold. Now BAP1 I showed you is important because it's mutated in about 85% of those patients who have monosomy 3 and go on to have metastatic disease. And what BAP1 normally does is it takes off a ubiquitin, as you can see here. So when it's blocked, ubiquitin is not taken off, and that keeps the DNA structure locked in to a certain conformation. And that conformation is obviously not a good one because it's one that encourages metastasis. But one interesting thing is that BAP1 is part of a complex that takes off ubiquitins from histones, represented by this arrow, but there's another complex, the PRC1 complex, and I should say this is the work of Bill Harbour at WashU, and this complex has a member BM1, or I1, and that member is actually regulated by the HDAC inhibitors, the histone deacetylase inhibitors. So this may be one reason that the HDAC molecule I showed you earlier, called SAHA, may have some efficacy, is that if it blocks this from being made, and this is part of this complex, and this complex drives the cell to put a ubiquitin onto the histone, you essentially can reverse the effect of BAP1. So again, we're not able to target BAP1 directly, but we may be able to target the effects of it being mutated. I showed you that. And so let me end by saying this. So our current state of knowledge is that mutant GNAQ11 and BAP1 loss are very important genetic drivers within uveal melanoma. I can say that we have exome sequenced many primary uveal melanomas. Exome sequencing means we've looked at every single gene in a tumor to see if it's mutated. No other genes than these three are mutated at that frequency. Yeah, that is 85%. Appear to be major mutant genes in uveal melanoma. Okay, I just repeated myself. Challenges. Direct inhibitors of GNAQ11 have not been developed. That's a major problem, but as I showed you earlier, that is being worked on. Loss of function mutations like BAP1 are difficult to target directly, but again, we may have ways of getting around that. Knowledge of the exact mechanisms by which these genetic aberrations work is incomplete. We have a lot of work to do in the lab. These are recent findings. The GNAQ and GNA11 story has only been within probably the last 3 years, and BAP1 within the last 1.5 years that we've even known that these are drivers within uveal melanoma. And single-agent treatment typically results in non-durable responses. This is just a theme within oncology, much less uveal melanoma, and therefore combination therapies will likely be necessary. But there are opportunities, and the opportunities are that we have one of the best predictive markers of future metastatic disease. When I tell my other oncology colleagues about monosomy 3 prediction or the gene expression profile class 1, class 2 prediction, they're amazed, because that is very rare to have that good a test for metastatic prediction. And what it's really good for is it allows us to treat the patients who are going to be at high risk and spare the patients who are likely not going to get metastatic disease. High frequency, small set of genetic aberrations, as I keep harping on, but this is very good too, because it means that for our trials, we'll have a homogeneous group of patients, and that really helps us. If you look at other diseases like lung cancer, for example, there's about a 3 to 5% ALK1 translocation, and then there's about 15% that have EGFR mutations, and then there's another small percent that have this, and then so you get this heterogeneous group of patients, and it's very hard to interpret the results of your studies. We have a homogeneous population, which really helps us in the interpretation results. Combinatorial targeted therapies may offer greater efficacy, and here's the real exciting one: combination of targeted therapies and immunotherapies. Why is this exciting? Because targeted therapies tend to give very marked short-term responses, that is, recurrence within 6 or 9 months or a year. But immunological therapies tend, if you do respond, tend to give longer duration responses. So it may be the case that we're able to use the targeted therapies that kind of kill the cancer, to kill cells, to release the molecules that are recognized by the immune system, and then use immunotherapies that enhance the effectiveness of the immune system by taking the brakes off to hone in on the cancer. And I'm going to leave the last one, just that we have a lot of other tools within the lab that will hopefully make our understanding of how these key genetic drivers work in this cancer better and do it faster and get it into therapies quicker. Thank you. Oh, I forgot. I'm sorry. I apologize. I should say we have a fantastic group at uveal melanoma at MD Anderson. Cmet Patel, who runs clinical trials along with Agat Badikian, Chandrani Chattopadhyay, and Elizabeth Grimm, who do basic science work. I do both. I see patients as well as do basic science work. We have 2 wonderful ocular oncologists, Bita Esmale and Dan Gombos. And then Dick Royal, who actually worked with Dr. Ping Pang at the NCI, came to MD Anderson, and he, as he mentioned, is going to start the liver-directed therapies at MD Anderson. This is my email, and I've been asked by a few folks if I have a card. I didn't bring my cards, but feel free to contact me if you have any questions or concerns. This is. Yes, and you're going to meet these folks in March because that's the next patient session. Yeah, great. Thank you.